Hello, and welcome to the latest Technology Networks webinar, Lab in the Loop, Smarter AI for Antibody Design. I'm today's moderator, Dr. Stephen Gibney, Science Writer for Technology Networks, and I'm excited to be here to host today's session. We have a fantastic presenter, Dr. Vladimir Gligorovich, who will be sharing some of his valuable insights with us. Vladimir is a Senior Director of AI and Machine Learning, leading a team of ML scientists and structural computational biologists who focus on large molecule drug discovery problems. His team focuses on developing AI and ML tools for optimization and design of both therapeutic and diagnostic antibodies, bringing the lab-in-the-loop approach to the large molecule drug discovery portfolio. After Vladimir's presentation, we'll have a short Q&A session. We encourage you to submit your questions at any point during the presentation. To do so, just type your question in the box on the right-hand side of the screen and click send. We'll do our best to answer as many questions in the time that we have available today. If you happen to encounter any technical difficulties during the webinar, click the chat box on the right-hand side of your screen to request assistance from our support team. Without further introduction, I'm now going to hand over to our speaker. Dr. Vlad, over to you.
Hello, everyone. Thanks so much for the introduction, and thanks very much for the opportunity to speak here today. So I'll be talking about our lab-in-the-loop framework for large molecules and how we use it to accelerate antibody design. First of all, let me tell you a little bit about why is it important to design antibodies and why antibodies are our molecules of obsession. So, of course, we have lab-in-the-loop in the company that we build for different drug modalities, but in this presentation, I'll be focusing only on antibodies. So on the left-hand side here, you could see the cartoon picture of an antibody. We often say it's a Y-shaped molecule. It's the molecule, like I said, of our obsession. This is the molecule that protects us from the world. It's generated by our immune response, our immune system, to block foreign pathogens. And on the right-hand side, you could see the statistics from the report from the last year of how many antibodies were FDA approved. And you could see that basically almost 20% of the FDA approved drugs are antibodies. In the previous year, it was even more. It was about maybe 30%. So they are one of the fastest growing class of drug molecules over the past decade. And that's mainly because they can be used to target very complex diseases, such as autoimmune disease and cancers, and as of very recently, Alzheimer's disease as well. For example, for cancers, one of the very first antibody designed by my company, by Genentech, is trastuzumab, and it was exactly used for breast cancer. The promising results of for example, lecanemab that are kind of reported a year or 2 years ago are showing that basically this antibody can effectively bind to the amyloid beta plaques accumulated in neural cells and basically can remove them and improve the clinical outcome of the patients. So the typical way to discover antibodies there are basically two ways. The first one is either to use some kind of library design and display technologies, and the other way that is very commonly used in my company is basically immunizing animals, and usually that's done by either let's say, having a specific target in mind that is implied in some disease. We know the pathway, we know the mechanism of action. Then we inject the target into animal, and then we let the immune system basically generate the antibodies that we then can test for other functions and properties. This is basically the mechanism that we use that is underlying this process. It's called VDJ recombination. Since I'm a machine learning scientist, the way I like to think about it is basically that our immune system is some very big generative model that can compute or that can actually give you a bunch of variants of antibodies. But basically, there are three different copies of three different segments in the of genes called V, D, and J that are basically these copies. They form three different types of gene segments are basically building the variable region of an antibody, and through DNA rearrangements, they cause one antibody of each of these gene segments to end up in a B cell. And as you can imagine, this sort of combinatorial space of combining these different gene segments can be very vast. And so we end up in something we call repertoire, which could be of the size of 10 to 11, 10 to 12 different antibodies that can be extracted from these immunization campaigns. These antibodies that we extract from immunization campaigns are very diverse in sequence and structure, which makes them very nice data set for training machine learning models. So that's basically the mechanism. So that mechanism, the VDJ recombination plus hypermutation affinity maturation, is the two processes that are basically generating a bunch of antibodies in these immunization campaigns that we are then extracting and testing for other purposes. Basically, a little bit about our team. So we are Prescient Design. We are, for a very brief period of time, we were a startup company that was acquired by Genentech. Now we're part of the much bigger computational department. We are called actually AI for DD, or AI for drug discovery. And broadly, we are interested in basically developing machine learning tools, making use of all available data across Genentech and Roche to accelerate drug discovery. So we do focus on other drug modalities. Like I said in the beginning, we do have lab in the loop for small molecules, for peptides, but basically in this presentation, I'll be talking only about antibodies. And this is very first problem that we were working on, or we were focusing on, when we first landed in Genentech almost 4 years ago now. And when we started this journey, basically we were given these three problems. One was called binder identification. The second one is binder optimization. The third one is de novo design. In the first problem, the goal is to develop machine learning models that can effectively mine these huge immune repertoires of the NGS sequences for the purposes of basically finding better hits and identifying new potential lead antibody molecules. In the second problem, we're often given an antibody. We also call it an antibody seed, that usually has some property we're trying to improve, or basically it's limited in some way. So for example, it could be that it has a low affinity and we want to improve affinity. And so the goal there is to develop machine learning models and use lab in the loop process to improve that property, or a range of different properties, in something we call multi-objective optimization process. And the third problem is the one that basically is now very, very popular. Many other companies are making a lot of announcements about this, and that's a de novo design problem. It's a very challenging problem. It's a problem which you're just given a target and maybe a location on that target, like an epitope, basically. And the idea is to develop an antibody from scratch, starting from target only. So no data available for him. At that stage, we don't have any data available for the model. So our mission is basically to transform the conventional drug discovery process by developing these machine learning tools that unite both wet and lab scientists in a thing we call lab in the loop that can be used to address these three different problems. So this is the way how we bring lab in the loop to a typical portfolio project. So like I said in the beginning, after you have a specific target in mind that went through the target assessment, that is implied, or that is basically part of some pathway or implies some disease, we do some of these discovery campaigns. So it could be immunization or something else, right? That gets us a hit. And then we apply lab in the loop to basically improve that molecule and optimize it for other properties, including affinity and other developability properties, by running this cycle of design, screen, analyze, and make, basically, process. So going into a little bit of details of this lab in the loop framework, basically, so the first component of our lab in the loop framework are these custom built generative models that produce antibody sequence designs. They can produce basically millions of different designs. These models usually start from some seed molecule, or in case if we run a de novo model, then you just can start from a target, basically. But if you start from a seed, you can then use these generative models to generate multiple different variants of that seed. Then designs are fed into a property predictor. And we have different kind of property predictors. These are things we call also pseudo oracles, and because they can produce or they can predict some properties of antibodies, including expression levels, non-specificity, binding affinity, etc. And these predicted properties are then used to rank designs in a thing we call active learning, such that we maximize the expected improvement compared to the seed antibody, right? After ranking, we decide on the number of designs we want to send to the lab. First, we do send the designs for to twist to get the molecules, to measure the expression, and then after that we can measure the binding affinity against the target using SPR or surface plasmon resonance, and of course we can do also additional functional screening and other characterization of these antibodies. The most critical part is getting the data back from the lab and feeding it back into the generative models and property predictor oracles, and such that these models can improve over iterations of lab in the loop. One important thing here that I want to emphasize is that, first of all, our approach definitely takes steps towards respecting important therapeutic constraints, because we optimize different antibodies for different type of properties, and you'll see that in a few slides. It demonstrates the general aspects because it can be, I'll show you how it works basically on different antigens, on different epitopes in these different disease programs on which we were running this lab in the loop. And it enables this autonomous antibody engineering. Basically, we can automate a lot of this stuff. The second thing I want to highlight here basically is this thing that is written right below the experimental characterization, which is the timeline. And the whole loop basically now, so that we get the results back from the lab in about 4 weeks with a lot of things with streamlined, and this whole process takes basically now 4 to 5 weeks, that the entire loop to get basically start from the seed and get an antibody. As you know, machine learning models can generate things from milliseconds to a few hours, right? We can get designs, but the most important part is making also the experimental characterization very fast, so that we can actually repeat or run multiple lab in the loops, basically. Now, I did mention in the beginning that there is an ensemble of different models, generative models that we use. And the main reason for that is because we don't want to limit ourselves on any particular model or limitations of one particular model. And so we want to really diversify the sequences in the beginning. And for that purpose, we basically use different types of models. Sometimes also these, what models we will use will be dictated by the project needs. And so in that way, we have a number of different generative models that can do unguided sampling, such as, for example, walk jump sampler or seq-PDM or some protein language models, or some guided multi-property optimization, such as a Lambo property enhancer and other methods as well. The other thing is that in many cases in a portfolio project, if we are given only the sequence, we run sequence-based models. If we maybe have a structure or maybe a co-crystal structure of a lead antibody or potential lead antibody with a target, then we can actually couple these generative models with other physics-based models, and we call these hybrid models. We also have a range of different structure-based generative models, and these are the models that we also use for de novo design, and these are such as, for example, FuncBind, Equifold, AB diffuser, etc. The reason why I'm mentioning all these names of the methods is because they are kind of published. We like to publish a lot in Genentech, and so we have a number of papers, machine learning papers, that came out in the last couple of years that are basically custom built, in the sense that we are basically designing our own methods always, taking into account the needs for each individual project and understanding a little bit the data and how we can model these complicated distributions of antibody sequences or antibody structures. So these are some of the papers here. There's, of course, the list is much bigger. If I have time, I will maybe focus on PropN, maybe towards the end of this presentation, and because this has been one of the most promising methods in our lab in the loop, basically with the highest binding rate and highest expression of antibodies that have been produced by this model. Of course, many of these models are part of this big publication that is right now in the late stages of a peer review process, and there we're focusing more on this lab in the loop procedure with all of these methods, and this is basically a huge collaboration between Prescient and antibody engineering in Genentech. And many of the results I'm going to show you here are coming from this paper, but if I don't mention something, you can certainly check it out there in this publication. So going a little bit into details of each individual component of our lab in the loop process. So like I said, the first component are these custom built generative models. And as you can see in the previous slides, right, we have actually a number of these different models. So what we do in every iteration of lab in the loop, starting from some specific targets and the seeds, what we do is basically we generate a bunch of proposals. We do restrict models on 30,000 potential designs, right? So each model, depending on how many models we have, we will end up with more designs, but basically each model is restricted to generate about 30,000 different possible candidates. Then these designs are, like I said, are fed into these oracles that are basically very useful because they're replacing very slow and expensive functional screening assays. So these are the models that are trained on the in-house data. Sometimes basically the models are coming from historic data from many different projects. And basically we then use this data to train these different kind of oracles. This combined library that comes from all of these different models is first filtered and ranked by our active learning framework, which is a very important piece of our lab in the loop. I will be mentioning that in the next few slides. But just to focus here on oracles, right? So we have oracles, sort of oracles that can test bind, no bind. These are basically classifiers that can predict the affinity level, or basically that can predict the KD values or dissociation values. It can predict the expression yield by some of our regression models. In addition to all of these oracles, right, that are measuring different properties of antibodies, we also have some quality control filters, and these are the filters that are checking for chemical liability motifs and things like that, things that we often want to eliminate if we see in the sequences. So variable region sequences are then synthesized as, so after we pass the selection and ranking, the variable region sequences are then synthesized as linear DNA fragments. And this is a sort of the job of Twist Biosciences, right? And so this kind of a linear DNA fragment-based expression workflow trims multiple cloning steps, saving time a lot and costs, while also being very amenable for automation. And after expression, antibodies are affinity purified, and concentration is measured by optical density, and then we basically binding affinity of these designs are, and lead antibodies is also is measured by surface plasmon resonance, or SPR. After we get all these measurements, like I said, the most important step is putting that back into the models and retraining and then repeat. Of course, designing an antibody with generative AI is I want to say easy. However, designing a therapeutic antibody is very difficult. Well, there is a whole list of different properties and the values for these properties that needs to be in specific ranges, such that the project requirements are satisfied. And a lot of time we're basically given a molecule with some in early stage or maybe even some late stage development that has some problems, right? It could be some affinity problems, or it could be some developability problems. So here you could see some number of these different characteristics that we're measuring in addition to expression and binding. So that could be off-target specificity, thermostability, immunogenicity, sub-association, high concentration properties, like say high concentration aggregation, etc. Right? So these are all very important to be measured basically and included in this lab in the loop, so that such that we can actually optimize these antibodies for multiple different properties. And this is where we basically need something called multi-property optimization. So how can this sort of lead to better molecules, right? So first of all, it can reduce time to clinic because we can optimize these things in a very early stages of the portfolio project. We can increase clinical success rate, right? For example, if our generative models are designing something that could be indicating for, let's say, high immunogenicity risk or ADA risks, right? We can immediately eliminate those, right? In the in these optimization steps, right? Also, with some of the generative models, de novo models, we can also target previously undruggable diseases, basically. Now focusing. So we cover the sampling part, we cover maybe the inference part, the lab part as well. Now, the most important part is also the active learning part. And so how are designs basically ranked? So first, we have some predictions from these oracles, and we use these predictions to rank the designs to maximize basically the expected improvement compared to the seed antibody. Also to ensure that the improvement of these models is that the models are basically improving in the next round. The data that we get from the lab is, of course, then reused and retrained, and then we hope to achieve that goal. So in many cases, actually, we're given an antibody where there is, for example, a problem, right? It could be the affinity, or it could be some developability issue. The important part here is basically that we optimize that antibody for one property that is problematic here, while maintaining other properties the way they are. For example, if you have a high affinity antibody, but it has some developability issues, the whole idea is that we use the lab in the loop or multi-objective optimization in a way that will introduce some mutations and changes to the antibody such that you will improve that problematic property, but you will never do that at the cost of, let's say, losing affinity, right? Now, as you know, maybe the drug discovery is not really done like that, right? It's actually done very sequentially, right? Where you start from a number of designs, and then you're checking from one property after another, right? And this problem introduces these issues later on that needs to be engineered out somehow, because you will introduce some problems into the antibody. For example, the antibody that initially didn't have high concentration issues, or can have it in some stage of the process if you really don't take into account all of these objectives and all of these properties at the same time from the beginning. So that's basically the way how we like to say that we like to stay at the Pareto frontier, right? Where we need to ensure that basically we are not making changes in a way that we are destroying other properties while improving some other properties, right? And so for that purposes, we use a black box optimization, Bayesian optimization with multi-objective function. And our acquisition function that I'll explain how we define is the one that we are basically here optimizing with this black box optimization, such that we stay at the Pareto frontier of this property space. So we want to avoid expensive experimental assays for designs that are strictly dominated. So basically, it's good or better in all respects at the candidate design, right? So that's basically what it means to stay at the Pareto frontier. So the acquisition function here that we use for these purposes is called expected hypervolume improvement. And this is the indicator, this is the thing we're trying to optimize from round to round. And here is a very easy way to explain this. So of course, this is a work by our colleagues from Prescient, Jiwon and Natasha. So these different properties, like affinity, expression, developability properties, are basically forming a space in which, if we put our designs, they can form something called hypervolume. So basically, this hypervolume is basically occupied by these different designs that we are getting from our generative models, and then we have these values of these properties from our pseudo oracles. So we need to, since we need to look at all the objectives at the same time, right? Here, this figure here shows just two properties, right? It could be expression and affinity, for example. And the blue area here is the one that is formed by added designs. So the two orange dots and the one blue one that are forming basically this hypervolume. In this case, it's a plane that we're trying to maximize basically when we are running basically lab in the loop process. So the way how this is sort of done is basically, for example, if in cases where we are running round 2 of lab in the loop, we might have, let's say, 3 designs. That's shown here on the left-hand side. So we have, let's say, 2 orange designs and 1 blue one. So how do we decide which one we want to measure? These 3 designs are forming the hypervolume that is shown here in blue. It's a blue area. But we know that our previous round basically is forming the hypervolume that is shown here in orange. And so by subtracting basically these 2 hypervolumes, we can see where this hypervolume is improving, or basically which designs specifically are contributing to improving or increasing this hypervolume. And you could see that that's only the blue dot here. And that's the one that will be basically selected for ranking and for further experimentation. In a very specific case of, let's say, just looking at expression or expression yield and, let's say, affinity or the KD values that we're getting from SPR measurements, right, you could see here, for example, the black points are the ones that are kind of improving maybe the hypervolume in the second round of this iteration. So this whole process, the mathematics behind it, the optimization is sort of kind of described in these 2 papers, and it's a great work by Jiwon and Natasha. So where do we believe that this multi-objective optimization can bring more value, the most value to the portfolio project? Like I said, the typical way to run drug discovery process is mostly sequential, and maybe, I don't know, a couple of years or down the road, or 1 year down the road, after you, I don't know, spend X amount of dollars, right? You realize basically that you don't have your lead molecule. And that's because, I don't know, you started from, let's say, a pool of antibodies that you discover from, like, let's say, immunization campaign. And then you selected some, then you, let's say, did humanization. Then there was a drop in affinity, then you try to improve affinity, but then you introduce other mutations that are causing some other problems, and then you're testing these other problems later on, and then eventually you might realize that you actually picked completely wrong clones from the beginning because they were not falling into the set of designable, basically, antibodies. So how machine learning can help in this process? Well, the first step is that we always want to run lab in the loop as early as possible in the drug discovery process. And we could use it, for example, because by the time we come to lead optimization and when there are these other problems, we might be very much limited by how much we can actually fix the antibody, right? Once all these problems are already introduced, right? So the whole idea is that we don't introduce these problems by just running AI and lab in the loop as early as in the discovery process as we can. And the typical ways of how we do this is by, let's say, diversifying the heat expansion step with our generative AI models, or by doing constrained multi-objective optimization and as early as possible, right? By checking all of these properties or running all of these filters or any kind of biochemical features that could later on be indicators of problems for some other biological or some other basically developability properties for antibodies. So let me show you some results now, basically. So over the years, we've been running this lab in the loop since we joined basically Genentech. We've been running this on basically multiple different targets. So here you could see some of the targets that we were testing this model on. Right now, lab in the loop is used in almost every active portfolio project. And instead of just maybe optimizing 1 or 2 properties, it's been used for actually optimizing multiple different properties at once. And what we observed is that this framework is very successful, is that over time, as you're running this thing over and over again, basically you are improving always the expression or the number of antibodies that will express. And of course, you're improving the number of antibodies that will bind. And of course, you're improving the antibodies that are better than the seed. And that's exactly what I'm going to show you in the next few slides. So for example, here for these 3 targets, we've been running, for these 4 targets that I showed you in the previous slide, we've been running lab in the loop. Like we had like 4 maybe iterations of lab in the loop. And then we measured how well lab in the loop does basically in each of these iterations, and basically how many times. So we introduced some metrics of success, right? So the first one, of course, is the improvement on the affinity, and this is sort of computed by delta PKD. So first of all, we compute the KD, which is a dissociation constant from the kinetics parameters of the SPR curves. Then we log transform this value. So everything I'm showing you here is on a log scale, basically. And so we have a PKD value of the seed molecule, and we have a PKD values of our designs. And so we can now measure all the improvements between our designs and the seed. And so, of course, we could also measure things like how many times or how many designs we have that have 3 times better affinity, right? And so on the left-hand side, the histogram that you see is basically measuring exactly that. So in each iteration, how many times did we design 3 times better binders? And you could see that by the iteration number 4, this number goes to 25, 27%, for example. If you look at the middle plot, right, this is showing basically from all of the designs, from all these targets and different campaigns, basically we're measuring the difference in PKD between the designs and the seed. And as a vertical line, a dashed vertical line here, you see the things that are 3 times better. Number 2 here indicates 100 times better stuff. Remember, this is in a log scale, right? So as you can see, not only did we manage to design a lot of antibodies that are 3 times better, but we are also good at designing antibodies that are better than 3 times, right? Also, we measure the expression yield, and also here it's the same histogram showing basically the difference in expression yield measured in milligrams, right, between the seed and the design antibody. And the dashed vertical line is showing basically the 0.01, let's say, improvement in milligrams between the seed and the design. As you can see, there's also designs that are worse than the seed that we design over the process, but we also were able to rank or filter those out as we are selecting things for the next round. Here, another very promising result, right? So on the left-hand side are actually different seeds from different targets. And you could see all the designs that have improved affinity, right? So only the designs with delta PKD higher than 0. And you could see that, for example, for some seeds, we are achieving things that are definitely 3 times better, 2 times better, but there are some cases where we design an antibody in just a few iterations of lab in the loop that are 100 times better in affinity. Also, you could see how this is sort of how these numbers are improving over rounds for different specific seeds. So here you have these 4 targets and different seeds, so different starting antibodies, and you can actually measure the maximum improvement in the PKD value over rounds, and you could see basically that this is improving beyond something that is 3 times better, which is again shown here as a horizontal dashed line. Another very important thing to check when we're running lab in the loop is whether we achieve the multi-objective optimization. So like I said, multi-objective optimization will constrain our designs to account for non-specificity, expression yield, for example, that can impact downstream characterization assays that require specific amounts of protein material, for example, and some in silico developability risks, right, that we're also running. So the way that we usually test this, right, so first of all, we do have models that can check for, let's say, non-specificity. These are the things that are coming from DD ELISA assay. And so this is kind of shown here at the bottom left panel. And so to estimate the risk for non-specificity, we actually run this BV-ELISA oracle. And the score that is above 0.1 would indicate a non-specificity risk. So we confirm basically that all of our designs that basically fall below that predicted threshold. For some of the designs, I think there was a subset of them, we were also able to engineer out some chemical liabilities while also maintaining our binding affinity. Another way we check whether we are basically in the range of therapeutic antibodies, right, is that we run something called therapeutic antibody profiler, or TAP. And this is basically just the way to basically get a lot of parameters from our design antibodies and compare them to the biochemical features and parameters from antibodies that are therapeutic antibodies that are, for example, already used or that went through the successfully through the clinical stages. And then we can look at the statistics, basically how far away in the distribution are our designs versus, like, let's say, therapeutic antibodies. And here, again, the plot on the top is showing exactly that we, or majority cases, for majority of our designs, are in the right range for therapeutic antibodies. I do want to focus on, in the next maybe 5 minutes, 10 minutes, on maybe one of our very promising method, and dive deeper into machine learning maybe a little bit. So as you know, for example, if you want to do some kind of conditional design, nowadays is the common practice is to do the guided design that consists of two parts, right? Say if you're designing a machine learning system, it should have two parts usually. One is the generative part, the model that generates proposals, and the discriminative model that checks for different properties, right? And can actually filter out designs that are not satisfying specific requirements. And so the most standard setup is to have these two different models. However, in most low data settings, these models are not doing the right job. For example, the discriminative model cannot be reliable if you don't have that many data points. So if you're working in a low data regime, which is often the case in many of these projects. The other issue is that these discriminative model that is guiding the generative model can basically guide the generative model in directions where there are no data points. So that's we call out of distribution of manifold. And of course, this discriminative model, if you don't have enough data points, cannot learn very complex distributions of these different properties, which is often the case for some of these developability properties. So we introduced to basically improve this, or basically to avoid having some problems like that, right? We introduced a model called PropN. Basically, we realized that having the discriminator being a bottleneck is basically the bottleneck for this, and we decided to find a workaround and completely sidestep this explicit guidance. And so we successfully do that by incorporating a technique that is basically coming from causal inference called matching. And this lets us train a discriminator-free model. So we only have one model here that is very suitable for low data regime. It doesn't propose designs that are from out of distribution. It has no problem with basically rigid property landscapes. It's very general approach, so you can actually use it not only for antibodies, but you can train a similar model for other drug modalities. And we call it property enhancer because it can explicitly learn to approximate the gradient of improvement. It consists basically on just these 3 steps. The first step is matching. And we split the data. Let's say we have a data of antibodies, like a bunch of antibodies with, let's say, affinity measurements. So we split the data in low property values, so the antibodies with low values of affinity and high values of affinity. And then for each antibody in the first set of the low antibody data set, right, we find the matching partner in the high affinity data set, right? So these are usually the antibodies with a low edit distance or that are very, very similar. But that have drastically higher values, specific values that could be affinity or something, or it could be also other properties as well. And we call this match data set. And then we move to the next step, in which we train some kind of an encoder-decoder model in which we map one antibody to another. And this implicitly learns the gradient of improvement. And then once you train a model in this way, you're actually increasing the number of data points, because you might have your data set initially could have some number of antibodies, but when you make the number of pairs, you actually are increasing the data set effectively, and you can train a model that can learn this directly of how to basically improve an antibody. Once you train a model like this, you can use it in an inference stage, where you just start from your seed molecule and then you plug it into the model, and then it's basically you can use it for sampling antibodies that are supposedly have the higher affinity, right? And then you can repeat this step by recycling. Basically, you can put the generated antibodies back into the encoder and then generate another batch of antibodies, etc., etc. And one very interesting step here is that basically, if you look at the table, in many cases, this method is generating really high binding rates and expression rates. For example, in 3 rounds of lab in the loop, we were able to basically get to something that is like even 40 times better binders than the ones we started from the seed molecules, basically. So this is a very interesting and promising method. We also have a publication about it, so you can read more about the technical details, the math behind it, and also the details of how we train this model and how we use it on some of the projects. Maybe last thing I want to mention is de novo. This is something we're also working on. I just want to say how this would be very transformative for basically antibody design. So right now, the procedure I described with lab in the loop that are used in lead optimization are basically that we, from a repertoire set, right, after we do immunization, we train some models, and then we use it for basically optimization and getting better binders. So we use it for binder discovery and optimization, basically these 2 challenges that I mentioned in the beginning. The de novo long-term goal, of course, is that we do want to start let's say, a portfolio project with just a target. We don't need to do the immunization or any library design, and we just run our de novo model with, let's say, a painted epitope for which we can design an antibody from scratch. And this could drastically increase the speed of generating antibodies and optimizing them, because we can then in that step also include all this multi-objective optimization. And so the way we think that de novo will enable antibody design is that, yes, it will be used for binder optimization and diversification, the same way we use lab in the loop. But we can start from an antibody scratch antibody, basically that is from scratch. We can design an antibody without any of the discovery campaigns. It could be used for binder identification, so we'll be able to generate antibodies, let's say, that could diversify for different epitopes. It could be very diverse, right? And that's very important for hypothesis testing and for other things as well. We could easily design different parts of an antibody and then assemble them into complex formats, and we can also run de novo for that as well. And yeah, we are hoping that this will drastically increase the time from a target to the lead molecule. And then there's a lot of ways we can do this structure activity hypothesis, because we can generate multiple designs. We can maybe even simulate some dynamics with it. And all of that can basically come from training these models. Maybe augmented with some of the MD designs as well, that can basically improve the trainings or increase the training set or improve these models. I'm going to stop here and just acknowledge the support from actually multiple different teams from Genentech. Actually, these are all the people. I mean, predominantly the collaboration between antibody engineering and Prescient is the one that was driving all of this. But of course, other departments are significantly contributing to building different parts of our lab in the loop system. Thank you very much for listening, and I'm happy to take any questions.
High-throughput screening is fundamental to biomedical research, particularly for the development of protein-based therapeutics. By iteratively screening protein variants, researchers explore a protein's vast sequence space in search of a novel variant with desired optimized properties. However, finding the right protein variant is difficult, even with the latest AI tools. Large-scale screens and training datasets for machine learning require pools of DNA, encoding thousands of defined protein variants to effectively sample the protein sequence space. There are many ways that researchers can generate their screening libraries, but current technologies leave much of the sequence space out of reach and underexplored. If your protein is longer than 300 nucleotides, your options for building screens are either very expensive, very limited, or very biased. As a result, the sequence space of most known protein sequences cannot be effectively studied in high throughput. To advance therapeutic protein development and AI-driven screening, researchers need tools to economically synthesize longer sequences at scale. At Twist, we constantly ask, how can we give more scientists access to longer synthetic DNA on a large scale? Our team used our proprietary silicon-based platform, optimized chemistry and automation to become the only company able to synthesize precisely designed pooled sequences that push the boundary beyond 300 nucleotides. This innovative new product, called multiplex gene fragments, consists of uniform pools of double-stranded DNA that are designed to your sequence specifications. In just 1 week, you can receive pooled lyophilized gene fragments, up to 500 base pairs ready for use in your high-throughput screen. With access to significantly more sequence space, you could precisely synthesize a library to generate training datasets to improve your machine learning model, or screen single-domain heavy-chain antibody variable regions and make discoveries that will lead to the next blockbuster protein therapeutic. So, what will you do with significantly more sequence space? Get started with multiplex gene fragments today.
Thank you everyone for your attention, and thank you, Vlad, for that incredible presentation. We're now going to move on to the Q&A session. Feel free to submit your questions using the box on the right-hand side of your screen and just click send. We've got just under 10 minutes, maybe 5 to 10 minutes, so we'll do our best to address as many questions as we can. We've already had quite a few questions, so if it's all right with you, Vlad, I think we're just going to dive straight into answering them. So the first one we've had are, what were some of the key challenges in integrating wet lab data with the machine learning models during iterative optimization?
Yes, see. So I would say there was a challenge before that. That was basically first gathering all the historic data so that we can have some initial set of models. And then basically using these models for the first basically version of the lab in the loop system. The challenges were basically the speed, right? We wanted really to run this really fast, get the validation of our models. So in the first, the first basically year of building this system, the challenges were mainly about like, how do we quickly test some hypotheses? How do we quickly do these things? And so there was a lot of improvements to streamline the whole process. There was also done like the experimental part, speeding up that a little bit of like, how do we submit these things, building the whole platform that gets to antibody engineering for further testings, and then it gets submitted for, let's say, Twist, and then back to the lab, and then measuring SPR. That process first was a bit longer, and then we streamlined the whole process, and we got like down to 4 weeks, which is amazing, I think. And then you can actually run this really fast right now. So I guess that was a few different difficulties that I guess were not solved by me, but my job was more like to basically filter out some of the good generative models that we could use here, get the initial data sets that we can run, that we can train these models on. And yeah, the linear DNA fragments that was done by some of the members of the antibody engineering in Genentech was significantly, for example, improved the time for getting the results back and doing all of these things. But yeah, so the challenges were always the speed, and it still is today. But I think a lot of stuff are automated since that first time we started doing this.
That makes a lot of sense. Again, a lot of work there. A lot of work went into it, obviously. So we've had a similar question in terms of it's on challenges. So someone's asked, what are the major challenges when applying AI models to predict bacterial resistance mechanisms?
I don't. Specifically, I don't work on that, like bacterial resistance mechanisms, right? So we test mostly stuff for antibodies, and these are completely different set of functions and properties we're sort of interested in. And so I cannot really answer that question. But there are different teams in Genentech that are working on this problem. I know that for sure. So unfortunately, this is not something I'm focusing on.
That's fine. It actually goes into potentially another question of what other projects do you plan or do you think that could use the lab in the loop system?
Oh, there are many projects. I mean, in the company that are using lab in the loop. It could be that there is a project that I'm maybe not that much of aware for bacterial resistance as well, but I'm pretty sure you could use it for that as well. Other projects, yes, in my group, in my team, we use it for small molecules, for large molecules or antibodies, and for, of course, peptides. And there is some projects on RNA design as well. So it's pretty much drug modality agnostic, right? So you set up generative models, you set up the oracles or oracles. You could have some active learning approach depending on, your just parameters will change. Your parameter space would change depending on, the field or whatever you're using. But basically, I know for sure that this whole concept is used in many, many different problems. In the company, we're mostly using it for drug discovery right now, and that's small molecule, large molecules.
Fantastic. We've had two more questions that are kind of linked here. So the first one is, what is the success rate of AI-derived antibodies from design through to clinical translation?
I think we are still in the process of evaluating that. What I showed you here is that you could improve an antibody. You could save a project, a portfolio project, right? That's been stuck in some stage where you want to, let's say, engineer out some part of the antibody that is related to some developability issues, right? So you could use this approach for that things, like for these problems, basically, specifically if you want to engineer out some things that are causing poor clearance or high concentration issues or stability issues or affinity, right? So you could use this approach for sure to improve these things. I think we'll take, so we do have some number of antibodies that reach I wouldn't say they reached the preclinical stage, but I think it's too early. I mean, the answer for this question is like it's too early maybe to evaluate this. But we have a number of projects that are basically in the stage where we're doing good with functional assays that are really well optimized, that we use on animals, and that still are doing well until we get the real feedback on, for example, some immunogenicity issues or some kind of ADA risks. It will take some more time to see how this will be done in the preclinical stages. But I would say the antibodies we developed are still kind of in the stage where we can measure the success only in this kind of a up until preclinical stage, basically. So it will take some more time to kind of see that, but we have a lot of ongoing work, like ongoing things where we'll get that feedback soon on how well the lab in the loop did in this case to improve the antibody all the way to the clinical development.
Well, one of the questions we've had actually is kind of a two-part, and you've answered it a little bit there. So someone says, how many real-world working drugs have been designed by Lab in the Loop? But obviously you've said it's still very early days. So the second part we've said is, what makes it so precise, this system? What is it that makes the Lab in the Loop so precise?
I'm not sure what it's precise in designing or improving antibodies. I like I said, the metrics we used here are when we look at have we actually improved that antibody. Mostly in the early days, we focused only on expression and affinity, and we wanted to see what are the limits of this, how much we can actually, with generative models and these oracles and active learning, we can actually, and a few iterations of Lab in the Loop, we can actually improve these properties. Of course, other things are also extremely important, and this is something we're constantly testing for, but with like low throughput stuff. And so I am not sure how still to measure fully success here. What makes it precise? There is, of course, explanations. I mean, maybe the question is why this is working. Or let's put it like that. Well, there are many reasons for that, right? It's the first of all, in each iteration here, like in most cases, you're dealing with a low data regime, right? And around that seed molecule that we want to improve, you don't have that many data points, right? And what Lab in the Loop helps you, it's not, of course, you don't expect to immediately design the best antibody. That's why we run this iteratively, right? But what's happening in this process is that you are collecting more data points around a very valuable seed molecule that you're trying to improve in these iterations, because you are measuring. And its important part is that these generative models and discriminative models are helping you push this in the right direction, in which you are making your models better and you are going in the direction to get an improved antibody, right? Because that's how these things are designed to, like with the guidance in the generative models, is exactly designed to go in the direction that you want, right? While you're also increasing data points and increasing the chances for the next round. So that's why I believe this is often leading to success, or to the examples that I showed you here are the ones where we really saw the improvement in antibodies, improvement in affinity, improvement in expression. And so, so these are some of the, I guess, the reasons.
Yeah, we've got time for one more question, so we'll just a bit more bigger picture. So just to finish on, how generalizable is the Lab in the Loop approach to other types of biologics or therapeutic targets beyond just antibodies?
I would say that's a very good question. And like I said, we do run separate Lab in the Loops for different modalities. I don't think you can generalize it for other modalities, but some models that are building blocks of this Lab in the Loop can generalize to other drug modalities, right? So you could have a generative model, or that is a de novo model that is trained on everything. Basically, it could be both large molecules, small molecules, etc. And then as it sees more data, it can become better and better, right? And you can run it for same model for both small molecule and large molecules, for example. But the very interesting issue here is, the problematic thing actually, but it's also interesting in the sense that it would be nice if it could be improved, is that some models, and that could be, let's say, property predictors or oracles, don't even generalize between different projects, even within the antibody space, right? And that's I would say a bigger issue here, because if I'm running this for antibodies, right, I need to train some models specifically for each project, right? And so we do have many models are actually generalizable, and you can use them for predicting different things, right? Let's say affinity and so on. But some projects are very different, and for example, functional assays depend really on the project or a disease program, on a lot of stuff, and you cannot use the same thing that you use for one project. It doesn't generalize well on another project. So there are some issues on generalizability within antibody space, but between different projects. But also there are some good stuff that are working well, and they can generalize, let's say affinity prediction, for example. Other things are, for other things you might need a sort of a different lab in the loop system so that you can actually start optimizing these things.
Fantastic. That seems like a perfect opportunity to wrap up. That is all the time we've got today for questions. Just remember that any questions that we didn't get to will be answered offline as soon as possible. You can also continue to ask questions even if you're watching this webinar on demand. And don't forget to download your certificate of attendance, which is available from the handouts tab on the right-hand side of the platform. Once again, a big thank you to everyone for listening. And Vlad, again, a big thank you for your time today. It's been really interesting talking to you. Thank you and goodbye.
Thank you for inviting me.